Very good morning, students. Today we are going to have the first lesson of metabolism and metabolic regulation, that is biosynthesis and regulation of fatty acid biosynthesis. And here, so this is an overview of the location of subcellular localization of lipid metabolism, and we can have one by one. Okay, that is, I'll show the laser pointer. Okay, this is the cytosol of animal cell, and this is a plant cell, and this is cytosol of plant cell, and this is a vacuole, and also the mitochondria and everything. So this fatty acid biosynthesis is a very, very important metabolic pathway, so that occurs in the cytosol, and the cytosol is responsible for NADPH production. Okay, that is the major component responsible for fatty acid biosynthesis. So that is from pentose phosphate pathway, that is you already studied HMP shunt pathway, and based on the ratio of NADPH and NADP plus, and so this fatty acid biosynthesis is favored. Okay, that means for fatty acid biosynthesis, NADPH level should be more when compared to NADP levels. And next one is isoprenoid and sterol biosynthesis, mostly in the early stages of the metabolic pathways that happens in cytosol of animal cells, and complete fatty acid biosynthesis happens in cytosol of animal cells and yeast cells. So coming to the other subcellular organelles like endoplasmic reticulum, so both the plant and animal endoplasmic reticulum so involved in the phospholipid synthesis, sterol synthesis, the later stages, not the complete one, the later stages only, and fatty acid elongation and desaturation. Elongation, you already know that the fatty acid synthesis, this will form palmitic acid. The elongation happens adding of additional chains. Only a limited number of carbons will be available for the palmitic acid. Further chains of 2 carbon each added to the fatty acid by elongation process. So this happens in endoplasmic reticulum. And the next one is fatty acid desaturation. This fatty acid desaturation occurs in endoplasmic reticulum. Desaturation means addition of double bond. Okay, that means that is saturated means all the carbon valency, all the 4 carbon valency is saturated, and if it is C double bond C, okay, that is called as desaturation. And next one is mitochondria. So mitochondrial role in plant and animal completely differs, because in mitochondria there is no fatty acid oxidation. It occurs in this fatty acid oxidation occurs in the peroxisomes. Okay, the peroxisomes and glyoxysomes are special bodies, subcellular organelles present in plant cells. So that is why the fatty acid oxidation, that is the beta oxidation, occurs in peroxisomes. But in the case of mitochondria of animal cells, we don't have animals and yeast cell, okay, not having that fatty acid oxidation location, that is like in plant cell, okay, that is the glyoxysomes and peroxisomes, including we don't have the chloroplast. So this mitochondria of animal cell, fatty acid oxidation occurs. Oxidation means a degradation of fatty acid by beta oxidation, alpha oxidation, and omega oxidation. And acetyl CoA production is happening. Okay, that is the reaction occurs in mitochondria only. The ketone body synthesis occurs in mitochondria. Fatty acid elongation, that means so this is also a part of elongation, that is endoplasmic reticulum, and the fatty acid elongation also occurs in mitochondria in animal cells. So coming to the plant cell, I already told that the mitochondria doesn't involved in the that is fatty acid oxidation. And here in chloroplast, the NADP H production and the fatty acid synthesis occurs in chloroplast. Coming to the peroxisomes, fatty acid oxidation till the end of the fatty acid complete oxidation, and the catalase and the peroxidase will be present there. So this is the overall mapping of all the metabolic pathways when compared to animal cell and plant cell, animal cell including the yeast cell. So this is the overall view. Based on that we can go. So next one is the malonyl CoA. So malonyl CoA is a major starting material for the fatty acid synthesis. It is a lipid, and triacylglycerol, phospholipid, and cholesterol, okay, all the synthesis needed by malonyl CoA, and synthesis and breakdown occurs in different pathways and catalyzed by different enzymes that takes place in different parts of the cells. The 3 carbon intermediate, the malonyl CoA, required for the lipid biosynthesis. That is why, okay, this is 3 carbon, 1, 2 and 3. And so we can see the biosynthesis of malonyl CoA in detail. So this is malonyl CoA formed from acyl coenzyme A and bicarbonate. This acyl coenzyme A synthesized by mitochondria. Okay, now this acetyl coenzyme A, okay, and bicarbonate, both are the precursors, and also both are the substrates for the formation of malonyl coenzyme A. So this bicarbonate donates one carbon dioxide. That carbon dioxide is attached with this acyl coenzyme A, so the malonyl coenzyme A will be formed. So nothing difference, okay, because this carbon dioxide is coming from bicarbonate ion, and actually this is CH3CO. CH3CO SCOA is acyl coenzyme A. So this attachment of carbon dioxide happening in this location, okay, that is this enzyme is involved, acyl CoA carboxylase. So that is very important enzyme. And this enzyme, okay, having 3 different domains. So one domain is a trans carboxylase domain. I'll zoom, okay, this one. I think so this is the trans carboxylase domain, and this is biotin carrier protein domain. And so this one is the binding domain of biotin carboxylase. Here this is the biotin, for example, okay, this is biotin carrier protein, so that carries biotin is a hydrophilic substance, and hydrophobic substance, okay, that is having a reactive molecule, okay, this is called as biotin arm. So this having a lysine side chain. So this biotin is not freely available, okay, this is carried by a protein. This is called as biotin carrier protein. This is really needed for starting of fatty acid synthesis. And one more thing, okay, this one in the form of action of acyl coenzyme A carboxylase, and this having, okay, the biotin carrier protein is a gray region. You can see the gray region, and the biotin carboxylase, which activates the carbon dioxide by attaching it to a nitrogen, okay, in the biotin ring in an ATP dependent reaction. This reaction needs ATP, that is energy. You can see here the ATP is needed, and this ATP, in the presence of ATP and bicarbonate ions, okay, this biotin carrier protein reacts with, okay, the enzyme. Okay, this is the enzyme. You can see here this biotin carrier protein binds to the biotin carrier protein binding region, and automatically this is attracted, okay, by the hydrophobicity, and automatically this domain is hydrophobic domain, okay, that is attached by the biotin carboxylase binding site. Immediately, okay, by the action of bicarbonate ion, okay, this bicarbonate ion, okay, may be dissociated to form CO2. Okay, this CO2 is coming and binding here to the nitrogen of so biotin arm. And now, okay, after the donation of, okay, this one, the ATP drives the energy, okay, that is ATP to ADP plus PI. So note the point here, ATP is not donating phosphate. So this is involved in the attachment of bicarbonate in the form of carbon dioxide, okay, to the enzyme, okay, that is biotin carboxylase. So next one is, so coming to the next reaction, the swapping. That means after the binding of carbon dioxide, okay, to the that is the biotin arm, automatically, so this will swap. That means the complete swapping of this region, biotin carboxylase region, to next enzyme, okay, that is trans carboxylase. So this trans carboxylase, okay, maybe that is a hydrolytic property, okay, that means so that will remove the acyl coenzyme A with the carbon dioxide. This is acyl coenzyme A with carbon dioxide. Now this acyl coenzyme A is incoming, and this acyl coenzyme A and carbon dioxide is going to be attached by this trans carboxylase. So the very simple, including the name of the enzyme, okay, denotes the function. Transfer of carboxyl moiety by the enzyme, okay, that is what is called as trans carboxylase. So next one is this one, that is biotin carrier protein. So that is now that is freely available. It is ready for another action. That means now the malonyl coenzyme A is liberated. Now this malonyl coenzyme A is ready to enter into the biosynthetic pathway of fatty acid synthesis. Okay, we can see that, okay, in the next one. Yeah, this is the next slide, okay, showing a very important enzyme, that is fatty acid synthetase. So this fatty acid synthetase is having two different types. One is found in the vertebrates, that is fatty acid synthetase 1, and fatty acid synthesis 2. There is synthesis 2 is found in the plants and bacteria in the dissociated system. You can see here the found in vertebrates. In the vertebrates, it consists of a single multifunctional polypeptide chain, and it is having 7 active sites for different reactions like in separate domains. These are the domains that are represented in different colors. You can see here the KS domain and MAT domain and DH domain, ER domain, KR domain, ACP domain and TE domain. So 1, 2, 3, 4, 5, 6 and 7 domains are available. So we can see one by one what is the meaning of KS and what is the meaning of MAT. So just you can see here, this is the 3D shape of the protein, the enzyme, the fatty acid synthase, and this fatty acid synthase or synthetase, both are same only, and this one is involved in the synthesis of fatty acid. You can see here the first domain is beta ketoacyl ACP synthase domain, that is called as KS domain. So this is the KS domain, and malonyl ACP CoA ACP transferase domain, that is a MAT domain. This is MAT domain, that is malonyl or acetyl coenzyme A ACP transferase domain. And the next one is DH domain, that is the beta hydroxyacyl ACP dehydratase domain, and enoyl ACP reductase ER domain. This is ER domain, that is this one, and ER domain, and the beta ketoacyl ACP reductase KR domain. This is the KR domain. And ACP is acyl carrier protein. This ACP means this is acyl carrier protein. It's not a domain of this one enzyme. It is not a part of enzyme. ACP is a protein. That is also needed for the fatty acid synthase function. Without ACP, there is no fatty acid synthase function. So next one is TE. TE means that is the thioesterase enzyme. So that part is involved here. That is the thioesterase enzyme will splice or cleave the synthesized palmitic acid product from the ACP domain, and the ACP is ready for another reaction, another subsequent fatty acid synthesis reaction. So this is ACP, and you should not think, what is the meaning of ACP, how it will be like that. This is very simple. A protein, ACP is a protein, and that will carry acyl group. So this acyl group is not easily carried by the action of some functional moieties are available. For example, here this ACP is always attached with pantothenic acid, and 4' phosphopantothenic is very, very important, and pantothenic acid is a very, very important coenzyme, and this one having the sulfhydryl moiety. This is SH moiety is there. So this is SH moiety. This SH moiety is a reactive moiety, and this moiety is going to be attached with malonyl coenzyme A. So that is why this is shown in the pink color. The malonyl groups are esterified to SH group. So this ACP attached with 4' phosphopantothenic acid and also with SH moiety. So this is a very active one. So this is the overall view of fatty acid synthase, the reactions, and maybe by seeing this one maybe a little complex, but actually very simplest one. And this is a very simplest form of flowchart given in some of the books, like maybe the bacterial prokaryotic system. That is the metabolism of bacterial or prokaryotic system by David White. It was taken from that book only. And here I will show what is the meaning of ENS, B, and what is ATP and all the abbreviations. And here within the brackets some of the things will be given. ACC, ABCD, A and ACC, AB. So no need to remember this one. This is the name of the gene responsible for the production of this enzyme. So no need to remember this gene name and all. So omit this one, and the enzyme name and all the substrates and the reactions and what are the cofactors involved and everything you should remember. It's a very, very, very important question for the examination point of view. And these are the reactions in fatty acid biosynthesis. The enzyme B is enzyme with the biotin. Enzyme means what is the name of the enzyme I told. So this is enzyme. So this FAS1, fatty acid synthase 1. So this is the enzyme fatty acid synthase 1 with biotin. Biotin is a carrier. Biotin will accept the carbon dioxide. Without biotin, the carbon dioxide attachment is not possible, and this reaction is driven by ATP. The same reaction, okay, just now, okay, we completed this one. This reaction I'm going to explain in this step. So enzyme with biotin, ATP and bicarbonate. So this acyl CoA carboxylase is the enzyme responsible, and that will combine enzyme with biotin with the carbon dioxide. This carbon dioxide from bicarbonate will be going to attach with B biotin, and ADP plus PA, okay, that is from ATP after the donation of energy, okay, this will be released, okay, after that. Next one is there is an incoming molecule, okay, that is acetyl coenzyme A. This is acetyl coenzyme A and acetyl CoA carboxylase. So this same enzyme, okay, that is acetyl CoA carboxylase. Now this is going to attach the carbon dioxide with this carbon, okay, that is why. So this CO2, you can see here CO2 and the hydrogen, okay, for the valency, the balancing, and remaining thing is CH2 COSCOA. And so this CH2 COSCOA is acetyl coenzyme A attached with, okay, the carbon dioxide COO, and this is called as malonyl coenzyme A. And this ACP-SH, ACP means just now I told, okay, this is ACP, acyl carrier protein, and in this SH group is very important for the attachment, and you can see here ACP-SH is denoted here. So the name of the enzyme is malonyl CoA ACP transacylase. So transfer of acyl moiety of this one, that is attached with coenzyme A, so going to be transferred to ACP. That means the coenzyme A is removed at the same time, and this malonyl coenzyme A and the malonyl moiety, okay, this malonyl moiety is going to be attached with ACP. You can see here COO and CH2CO. So this SCOA is liberated, okay, by the transacylase, and in the place of, okay, this one ACP will join in the form of ACP SH moiety. The joining through SH moiety, okay, we already discussed, and this is already reacted with SH. That is why, okay, this CO reacted with SH of acetyl coenzyme A, the coenzyme A moiety. Now the coenzyme A is liberated, the same SH moiety of ACP is reacting with this one. That is why ACP with malonyl moiety is formed here. So next reaction is a very, very important reaction. This is a beta ketoacyl ACP synthesis 1, 2 and 3. So don't remember all the gene names and all. So beta ketoacyl ACP synthesis 1, 2 and 3. There are 3 subsequent reactions are there. Okay, the first one is here. This is acetyl coenzyme A, and this acetyl coenzyme A is not active form. So only with attached with ACP, okay, that is active. So the preliminary stage of, okay, this reaction, already the preformed ACP with the acetyl moiety is ready for the interaction with the beta ketoacyl ACP synthesis 1, 2, 3. So this one is reacting with this one, and the acetyl moiety going to be transferred, okay, to the CO moiety. You can see here just a simple one, ACP, the same ACP is there, and ACPS moiety, okay, that is already there, and the CO, CH2 and the CO is already there, and CH3 is going to be attached, and the carbon dioxide is going to be removed. Okay, this carbon dioxide going to be removed, that is why this carbon dioxide is liberated. So instead of this place, the remaining is CH2CO ACP. The CH2CO ACP is there, and to this, so this CH3CO is going to be attached. That is why, so this CH3 and this CO, and remaining part is after the removal of this carbon dioxide, remaining part is CH2CO ACP. This is CH2CO ACP. So now another reaction, there is a beta ketoacyl ACP reductase enzyme reduction. Beta keto acyl ACP reductase enzyme. This reduction, okay, by the cofactor or coenzyme, okay, that is NADPH. This NADPH is a very, very important one, and this is produced by HMP shunt pathway only. And without this HMP shunt pathway, there are 2 significant properties of HMP shunt pathway. One is NADPH production, and second one is 5-carbon compound, okay, that is 5-carbon sugar formation, okay, that is a pentosis. So all the nucleic acid bases attached with the sugar, okay, that sugar is called as pentose, okay, the ribose is a sugar. either in the ribose or deoxyribose based on DNA and RNA. DNA has maybe deoxyribose and RNA having ribose. So this NADPH is coming from HMP shunt pathway, and in the plants that is from chloroplast. Okay, this one. Next one is the beta ketoacyl ACP reductase. The reduction is going to happen, attachment of two hydrogen, this hydrogen and this hydrogen. So which point means you can see here this CH3 is CH3, the same intact will be there. So this CO moiety, so the CO moiety of acetate moiety, okay, this acetyl coenzyme A is COA moiety. This CO moiety we are going to attach CH, one hydrogen here, and O another hydrogen is here. Just you can see CO will be converted to CHOH. That's the only difference. Okay, and after the donation of NADPH plus H plus two hydrogens, and remaining fragment is NADP. So that is why NADP is liberated now. So next reaction, beta hydroxyacyl ACP dehydratase. So this dehydratase means the removal of water. So water is removed from where means, okay, from here. So that is, this is one hydroxyl moiety and another hydrogen is from here. So if you remove one hydrogen from CH2, that is CH, okay, there, and another hydroxyl moiety, so this hydroxyl moiety going to be removed and remaining portion is CH3CH. So that is why CH3CH double bond CH, because CH single bond CH will not be formed, because to balance the valency, because carbon having 4 valency, you can see here this carbon having 2 valency, and this also, and this carbon, this hydrogen is also 1 valency, and another valency shared with another carbon, okay, here, okay, and this one is the product. And next, enoyl ACP reductase, again reduction, and again the same reduction, okay, by NADPH. So this NADPH plus H plus, that is also HMP shunt pathway, and now it will reduce. The hydrogen is going to be attached here and also here. Now it is converted to CH3, and this is CH2, and another CH2 is missing here, students. Don't forget to add, okay, another CH2. So including in the book, okay, this is missing, and I keep on saying, okay, this correction, okay, in each and every class. So this CH3 is CH3, and addition of one hydrogen from this one to this one, okay, that is CH2, and this CH again converted to CH2 here. So this CH2 you have to add here, and remaining portion is COACACP, and the remaining part is NADP. So now this is the first stage, the first cycle of fatty acyl synthesis system, and this will continue for chain elongation. Okay, this is going for chain elongation, and in this case, okay, this will be the entry point, not this one. Okay, this is the entry point, that is CH3, CH2, COACACP is the entry point here, and that is going to attach with, okay, this one a subsequent way. So this will form C16 ACP, and C16 is palmitic acid that will be liberated. Next, coming to the regulation of fatty acid synthesis, how the fatty acid synthesis is regulated, because your subject is metabolism and metabolic regulation, and you should study about the regulation also, how it is perfectly regulated, and what are the key parameters involved in the regulation process, which one is the positive modulator and which one is the negative modulator. You can see here this is the overall map of the regulation part, and the green color indicates that is activation, and the into mark, the red color mark, indicates inhibition. So this is the pathway, that is citrate to acetyl coenzyme A, and acetyl coenzyme A to malonyl coenzyme by the enzyme acetyl CoA carboxylase enzyme, and malonyl coenzyme A to subsequent SEPs to form palmitoyl CoA. You can see here that we can take the end product. The end product is palmitoyl CoA, and so this palmitoyl CoA, okay, that means C16 number containing fatty acid, and this fatty acid is the first product of fatty acyl synthase 1. This is the product. If this product is more, the concentration is more, automatically you already studied a concept of feedback inhibition. If already pre-synthesized palmitoyl CoA is already sufficient, or maybe sufficient quantities are there is no need to synthesize the further amounts of palmitoyl CoA. So that is why this product will inhibit this enzyme. That is, the enzyme is acetyl-CoA carboxylase enzyme is inhibited. So that is why this will not form malonyl coenzyme A. So this is the first step. Second one is by the hormones. We have two hormones, and mainly insulin and glucagon. You know that insulin is very important for the carbohydrate metabolism, and also glucagon. You can see here the insulin triggers the activation of conversion of citrate to acyl coenzyme A, and so this acyl coenzyme A once formed means that will go subsequently for the synthesis of malonyl coenzyme A, and palmitoyl-CoA is synthesized. That is why we are reducing our glucose concentration in the blood circulation by converting all the excess citrate to palmitoyl-CoA, and all the palmitoyl-CoA is converted subsequently to the saturated elongation or unsaturated fatty acid formation and deposited in all the different parts of the body, especially in the adipose tissue. And that is why this insulin controls the glucose molecule. The excess glucose molecules will be converted into palmitoyl-CoA, and subsequently this will form cholesterol, or otherwise other molecules, linoleic acid, linolenic acid, arachidonic acid. All the further fatty acid molecules are formed. Second hormone is glucagon. Glucagon is the inhibitor of this one. That means if the glucose concentration is less, it has to produce glucose from non-carbohydrate sources. That is why further citrate will not be metabolized into malonyl coenzyme A. This is blocked. If the glucose levels are very less, and if the glucagon is induced at the time, this will inhibit this reaction. And the epinephrine trigger phosphorylation and also the inactivation of this enzyme. So next, coming to the induction of other parameters, including the substrate. Citrate is a substrate, and this will induce this enzyme. So this is induction. Induction means activation. So the citrate is a substrate that will induce more activity of this enzyme, acetyl-CoA carboxylase. So subsequently this will form more palmitoyl-CoA. So this is the basic steps involved in the regulation of fatty acid synthesis. You can see an example of acetyl-CoA carboxylase enzyme, and this is the 3D shape of acetyl-CoA carboxylase enzyme. So present in the membrane. There is you can see the 400 angstrom. That is the size of the enzyme. This is quite the multi-enzyme complex, and we already studied this one here. This is the complex. So this one is shown very clearly here. So acetyl-CoA carboxylase is activated by the changes, and the Mg plus, Mg2 plus, and the pH increase that accompany the illumination process. This is regarding the regulation of fatty acid synthesis and the routes of synthesis of other fatty acids. Now we are going to see how other fatty acids are formed from palmitic acid. Initially, the first step is the fatty acid produced is palmitate, 16 carbon palmitate. And here we are going to depict the number of carbon atoms and number of double bonds, and so this within brackets there is a delta 9 will be there. The delta designates the location of double bond. For example, here we can go with one by one. So 16:0 means 16 carbon palmitic acid, but there is no double bond. That is why 0 means there is no double bond. So by elongation, this will form stearic acid. This stearic acid, 18 carbon, but there is no double bond. By again desaturation. Desaturation means addition of one double bond. That is why the double bond at only one double bond, and if it is 1 means you have to represent that one, that is delta 9. Delta 9 means the 9th carbon to 10th carbon, that bond is a double bond. So delta 9 means, don't forget, that is not between 10 and 11, that is between 9 to 10. And further desaturation, that will form linoleic acid. This is linoleic acid. The two double bonds are there at delta 9 and delta 12, that is 9 to 10 and 12 to 13. So this is the indication. And further desaturation, the plants only the plants will produce linoleic acid, linolenic acid. That is 18:3. The double bonds and delta 9, delta 12, and delta 15. That is 3 double bonds are there. And delta 9 means 9 to 10, and 12 means 12 to 13, and 15 means 15 to 16. So from this, other polyunsaturated fatty acids are formed, and the linoleic acid, further desaturation will form gamma linoleic acid. So that is 18:3. That is 3. That is the double bonds are there. Delta 6, delta 9, and delta 12. By further elongation, eicosteric acid will be formed, and this is a very important compound in the inflammation and also the controlling the inflammation processes. And 20:3, 20 carbon and 3 double bonds. That is the double bond in delta 8, delta 11, and delta 14. And by desaturation, this will form arachidonic acid. Arachidonic acid is a very important product of the fatty acid synthesis, and total carbon atoms are 20, and 4 double bonds are there: delta 5, delta 8, delta 11, and delta 14. So this palmitate is a precursor of the stearic acid, and all the palmitate is the starting material of all the fatty acids. And with the further saturation, further elongation that may happen in smooth endoplasmic reticulum, and mammals cannot synthesize linolenate and or linoleic acid. So that is why it is called as essential fatty acid. Both linoleic acid and linolenic acid, both are not formed by mammals. So that is why it is called as essential fatty acids. It is required for life. So this is the unsaturation reaction. There is unsaturation means a removal of that is 2 hydrogens by NADP to NADPH. This is saturated fatty acid. Just see here, this is CH3 and CH2 n number of times, and finally CH2 and COA. So this fatty acid coenzyme A desaturase. Desaturase means that will form CH2 single bond CH2 is converted into CH double bond CH. This is called as monounsaturated fatty acid CoA. In this case, there are cofactors are involved. The first one is NADPH plus H plus. So this will convert this cytochrome B5 reductase into, that is FAD to FADH2. Now the FADH2 is receiving 2 hydrogens from NADPH plus H plus, and FADH2 is a very, very important parameter. This is a component of electron transport chain. That is the second complex of electron transport chain, and this FADH2 further converted into cytochrome B reductase by FAD, and donating 2 hydrogens to cytochrome B5. So this 2 cytochrome B5 is converted to 2 cytochrome B5, this Fe3 plus 2 Fe2 plus. So this Fe3 plus 2 plus by FADH2, there is 2 hydrogens used for the further that is oxidation process, and this one is Fe3 plus 2 Fe2 plus, and this component involved in the fatty acid CoA desaturase process. So this will receive 2 hydrogens from here and converted to Fe3 plus, and this is a cyclic process, and automatically done by the NADP and FAD and Fe2 plus and Fe3 plus substances. So that is where the first reaction, this saturated fatty acid CoA CH2 single bond CH2 converted into CH double bond CH. So this is the basic principle involved, and this occurs in south endoplasmic reticulum, and unsaturation reaction is catalyzed by the fatty acid CoA desaturase. Fatty acid CoA desaturase is the mixed function oxidase enzyme, and the fatty acid and NADPH simultaneously undergo 2 electron oxidation. So 2 electrons are involved. That is why the hydrogen is involved. The 2 hydrogens are donated by NADPH. So for this reaction is also NADPH is very important for the that is the desaturation process. Right. So with this we can complete all the biosynthesis and the regulation of fatty acid synthesis from palmitic acid and palmitic acid to subsequently other the fatty acid synthesis.